So today my presentation will be about molecular testing for acute myeloid leukemia, and I did like an overview over this article. So I'm gonna start. The next slide. Begin with an introduction about acute myeloid leukemia. It's basically a malignant neoplasm of the myeloid cell lineage, which is affecting blood and the bone marrow. It's a type of cancer in which the bone marrow starts to produce a large amount of blood cells. The molecular changes that come with the acute myeloid leukemia helps us to classify, to diagnose the disease, and in the prognosis and the treatment plan associated with the disease. And based on these changes, the WHO classifies AML into several types, including those with recurrent genetic abnormalities, therapy-related neoplasm, unspecified AML, and myeloid sarcoma. Did you hear me? I think so.
Yes, perfect. I can hear you very well.
Okay, now coming to how does it occur, there are two ways in which AML could occur. It could be either due to the chromosomal rearrangement, the translocation between chromosomes. For example, we talk about translocation between chromosome 15 and 17, chromosome 18 and 21, or the inversion within the same chromosome. In this case, it's chromosome 16. Identifying this translocation is critical for the diagnosis and the treatment of the disease because, for example, a translocation between chromosome 15 and 17 could lead to the formation of a fusion gene, the PML RARA gene, which is when we have two genes, each of them is functional, they fuse together, they retain both of their function, but sometimes their function will be activated more and leading to, the prohemolytic leukemia. The second cause would be the AML with a gene mutation. It's when we have a mutation only in a certain gene. For example, the NPM1 gene, the CEBPA gene, which is responsible, for example, for a transcription factor, and the RUNX1 gene. Detecting this mutation is also important for the diagnosis and the treatment of the disease because, for example, FLT3 mutations indicate poor prognosis, means the patient is not able to recover without having the treatment, but it also allows us for targeted therapy by using the FLT3 inhibitors. Now, coming for the part of how do we test the molecular testing for the AML, we could use karyotyping, fluorescent in situ hybridization, PCR, traditional sequencing, next generation sequencing. All of these methods help us to identify the DNA changes, whether translocations or mutations that are occurring, and will help us to diagnose that disease. The first one, of course, it would be the clinical pathogenic assessment. Of course, we will be assessing the symptoms that are appearing on the patient, like fatigue, bleeding, anemia, while pathogenic evaluation focuses more on the bone marrow and the blood myoblast counts, which are typically high. Typically, 20% of the blasts in the bone marrow or the blood are used for diagnosis, but molecular changes can lower this threshold. That's why the guidelines recommend karyotyping and FISH for AML patients with various methods used for subclassification, risk stratification, and treatment decisions. Sorry, we don't have electricity now. Anyway, for the second method, it's karyotyping. Karyotyping is a cytogenetic technique. It's widely used to analyze the chromosomal abnormalities, which help us to identify AML and its type. In karyotyping, we culture the cell, we arrest them at the metaphase stage, and then we stain them and we visualize them, we arrange them in order. And that's how we could detect the translocations or the inversions in the chromosome. However, this has a limitation because we require cell proliferation and the inability to detect small or cryptic chromosomal changes. And the cryptic chromosomal changes are those, we call them hidden changes in the chromosome that sometimes mainly occur in the regions, the entrance region, which are not detected by such technique and require other techniques, which I'll talk about it, which is the fluorescent in situ hybridization. So in fish, what happens is that we use DNA probes, which are complementary to the gene of interest, and when these they will hybridize, and we will label it with a fluorophore. So it will, when it hybridize, it will fluorize, and we will detect where exactly the translocation or the mutation happens. And this is way more specific to detect the cryptic changes. Now, the fourth method is actually PCR. PCR is actually a way we amplify the DNA of interest, and it's very specific because we are using certain primers and probes, which are short oligonucleotides, 10 to 30 base pairs. And when we amplify them, and if we are targeting a certain gene of interest, so if it's high, depending on the CT, we can decide like what's happening. And however, it also faces challenges such as requiring multiple primer pairs for translocations with numerous breakpoints, which can reduce the practicality for certain translocations. The other method is Sanger sequencing. Sanger sequencing is also a traditional method to detect point mutations, small insertions, and deletions with a detection limit of approximately 15% allelic frequency. And Sanger sequencing depends on the concept of dideoxynucleotides, where there will be a chain termination when the nucleotide is there, and it's also specific. There's another type of sequencing, which is the pyrosequencing, that have higher sensitivity, around 5%, but it's limited to detect shorter DNA sequencing, while Sanger sequencing covers hundreds of pairs. It could be up to 300. The other method is next generation sequencing. It's the comprehensive genomic analysis. It's capable of sequencing hundreds to thousands of genes, sometimes the entire human genome in one test. It could be either the whole genome sequencing, where we are sequencing the introns and the exons, like everything, or the whole exome sequencing, where we are just sequencing the disease-specific gene panels, the exons, the genes which are expressed and they are commonly of interest for us. NGS panels with high sequencing depth can detect variants with low allelic frequencies, around 5%, and it could be used for both DNA and RNA. Now, for other molecular testing technologies, there are several other technologies. For example, we have spatial transcriptomics, which combine molecular analysis with microscopic localization, allowing for the visualization of molecular changes within specific anatomical structures or cells. We have circulating cell-free tumor DNA, because the tumor DNA sometimes would be the tumor cell is going to like disseminate the tumor DNA into the blood. So we can do tests in order to identify these tumor cells in the blood circulation, which aids in the prediction of tumor metastasis and recurrence. Although primarily used for solid tumors, these technologies may also be applicable in detecting the molecular changes in AML and in the hematopoietic microenvironment in the bone marrow, for example, which is the case of AML. Now, here I'm going to be summarizing some examples of the ones that the author did in the article. The first one is the APL, acute premyelolytic leukemia, which is the translocation between chromosome 15 and 17. As I said, it will lead to the formation of the PML RARA chimeric gene. And the chimeric gene, what differs from a fusion gene is that in the chimeric gene, we have portions of the two genes that are going to combine, form a new function. While when we have a fusion gene, it's just the two genes combined together, each of them retaining their own function. And this APL is linked to disseminated intravascular coagulation, but responds well to triatonin treatment. The translocation between chromosome 15 and 17 is typically identified through karyotyping, and faster and more reliable diagnostic methods include FISH, of course, it's more specific, such as the dual color fusion FISH and the RARA dual color break apart FISH assays, which identify various translocation, including this translocation. PCR assays, particularly a real-time quantitative reverse transcription PCR, here we may use the RNA, are also utilized due to their higher sensitivity for detecting the cryptic translocations, which are the hidden translocation mean. Now, the second example is the AML with the translocation between chromosome 8 and 21, which results in the formation of the RUNX1 T1 chimeric gene on the chromosome 8, and it's often identified by karyotyping. However, for cryptic translocation or non-proliferative cells, fish assays, specifically the dual color fish, are used for more sensitivity. And AML with this translocation typically has high remission rate and favorable prognosis. Now, the third example is actually the AML with CEBPA mutation. This gene is responsible for coding for a protein, which is a transcription factor that will regulate the expression or the inhibition of a gene and how much it will be overexpressed or underexpressed in a cell. It's found in 4 to 9% of younger AML patients, and CEBPA mutations are best detected using sequencing methods due to their potential occurrence throughout the gene. Sanger sequencing is the gold standard, but it has 15 to 20% detection limit for variant allelic frequency. NGS is also employed, although its effectiveness can be hindered by the high GC content. Hybridization capture NGS assays are preferable over amplicon assays due to better coverage and accuracy in sequencing this gene. Now, at the end, they talked about the clonal evolution of AML, which is when the tumor environment or the tumor itself changes over time. It like maybe due to mutations or these point mutations. These will lead to the change in the microenvironment and hence evolution over time. And the next generation sequencing assays are the ones which are effective to track this clonal evolution, with single cell NGS providing even the more detailed information. However, they propose certain future improvements that should happen in the NGS to help in the molecular testing of AML. The first being to expand the gene coverage. So research should discover more genes so that we could sequence them and discover them more. And how they are related to AML. Enhanced sequencing for different difficult genes. When we talk about the cryptic genes or large insertions, deletions, and the challenge of high GC content that affects the results. Improved non-coding region sequencing, recognizing the significance of the intron sequences, and especially for the genes linked to the AML. And finally, they recommended to have better translocation detection, refining methods to detect various translocations. Those were their future improvements, and that was it.